we are looking at genetics of blood type. And so that means the blood types A, B, AB, and O. And this is a case of what's called multiple alleles. Instead of just two genes, a dominant and recessive gene, we have more than two genes. And they are shown here. There's a capital I and a superscripted capital A. A capital I and a superscripted capital B. And a little I right there. Those are the three alleles or genes for blood type. What we see is A and B are codominant. If they're both present, both traits are expressed. But these both dominate over the little I. So they're dominant over that recessive gene. These are the proper symbols that we use. Don't invent your own symbols because then you'll get a lot of things wrong as we go through. So now what we're going to look at is the possible combinations of these. So we have three alleles and we're going to see what are all the different ways that we can combine these things. Well, if you have blood type A gene, you are either homozygous dominant, two capital A's, or you have the capital A and the recessive gene, you're heterozygous. Well, if that's true for type A, type B has to be the same kind of thing. Type B is either homozygous dominant, the two B genes, remember the capital I's, or heterozygous like so. On the other hand, if you're type AB, there's only one possible combination, the A and the B. Codominant because both of those code for the antigens we find in the red blood cells we talked about in the last video. And you can tell with type O, you're going to have the two recessive genes. So here we have 6 genotypes, but we know there's 4 phenotypes. If you're type AB, that's your genotype. If you're type O, little I, that's your genotype. But if you're type A or B, you need more information to know if you're homozygous or heterozygous. So all of this information is summarized in this little chart here. So now we look at the genetics of blood type, and you're going to have to do Punnett squares, and you're really going to have to think about what's going on. So we said, hey, what happens if we take two heterozygous, one type A, one type B, and we do a cross? What kind of things can we get? Well, we go IAI, and put that there, and here's our IBI, and we put that there, and we say, hey, these are the possible gene pairings. And this is a fascinating cross because from this, we had 2 different types of blood, but we see all 4 blood types are possible from that cross. It's a 25% chance of each blood type. And this is certainly not going to be the case for other combinations. And you can try some of your own. I haven't said if it's homozygous or heterozygous. You can decide there. And you can try a type AB with a type O. So that's what this space is. Try it out. And if you're having problems, send me an email, ask questions. But that's the genetics of blood type. Make sure you're using these proper symbols. These are the phenotypes on the left. These are the genotypes on the right.